My name is Mary Matoes. I am a research tech at the Center for Applied Genomics, working under Dr. Halkan Halkan-Arsen and Dr. Marina Bukai. And I am the one working on, thank you. And I am the one working on the mouse model and phenotyping the mouse model via behavioral studies. So some background and our experimental approach. As Dr. Bukai said, we received the mice from UC Davis. They were on a mixed background strain. Now, for behavior studies, it is very important for the mice to be on a single strain. It should not be on a mixed background strain. So what we decided to do to mitigate any possible gene interactions between CHAMP and other genes from the mixed background, we bred the CHAMP1 mouse line into the C57 black 6 background. This background strain is very good for behavior. It has shown in literature that it's one of the better backgrounds for behavior. So what we did is we did 4 generations into this background before starting any behavior studies. These litter sizes range from 5 to 10 mice per litter. It's usually 50% males, 50% females, with 25% of the mice being heterozygous for the CHAMP1 mutation. We have preliminary experiments that were performed on mice bred on the 4th generation, so about 95% into the C57 black 6 background strain. We start behavior around 8 to 10 weeks old. And here are the timeline of behaviors. So day 1 was elevated zero maze. Day 3 was open field. Day 5 to 7 was accelerating rotor rod. And day 9 is acoustic startle with pre-pulse inhibition. I will go through each of these behaviors in the coming slides and explain them more in depth. So, first one, elevated zero maze. It is a very common test for anxiety-related behaviors and fear behaviors. So what you do is, it is that circle platform right here. You place the mice in the closed left portion of the apparatus, and it is a 5-minute trial. So you literally just let them walk around the circle for 5 minutes, let them explore. Mice naturally like to spend more time inside the walled quadrants instead of the open quadrants because they are very naturally fearful animals. So when other papers did like anxiolytic treatments, the mice actually increased their time in the open spaces. For our CHAMP mice, we did not see any differences between the groups. They acted, as fearful as any mouse would act. Now, spontaneous open field activity. It is a commonly used test to show any hypo or hyperactivity in a mouse. So a lot of just walking, ambulatory behavior, and making sure that they're able to walk around in a normal manner. So they basically, you put the mouse inside of this chamber, and there's infrared beams that capture when the mouse is either walking in the center, is walking on the perimeters, or is rearing, which rearing is they put their little hind legs up and they're kind of just like looking around, exploring. The data that is shown when you get the results is measures of the distance that they traveled, their rearing bouts, like I said, and center activity, so how long they spend in the center of the chamber. As you can see here with these 3 results, all of the wild type and CHAMP mice seem to have similar ambulatory behaviors about exploration, which is a good sign because then we can do more complex behaviors, like I will show you next. Rotarod. Now, rotarod is a motor activity behavior. So what we do here is there's 5 little walled rods. We each place one mouse on the rod, and it accelerates from 4 to 40 RPM. Now, they receive 3 trials a day for 3 days. So what we're supposed to be watching for and seeing is their cadence. Are they able to stay on the rod without falling or rotating 360 degrees around the rod? So we measure the time to fall or grip is recorded as a measure of their coordination or their balance. So the initial cohort of mice that we had, weirdly enough, the heterozygous males and females, well, trend in females, show an increase in being able to stay on the rotarod, which we were thinking about. We're like, what if that maybe suggests a repetitive behavior? A lot of the time, a lot of papers, literature that we've seen, they, when a mouse, specifically in autistic models, or have an increase in latency to fail, it usually shows repetitive behaviors. So we decided to adapt to a different version of the rotarod to investigate if it will still show an increased latency to fail. So we did high-speed rotarod. So high-speed rotarod is very similar. They receive 3 trials a day for 4 days now instead of 3 days. The first 2 days is the regular 4 to 40 RPM on the rod. Days 3 to 4 are 8 to 80 RPM, so a little faster. Like I said on the previous slide, some IDD-related models show better performance than the wild-type littermates, which can suggest some repetitive stimming behaviors. However, unfortunately, we ran a cohort of this, and they really didn't show any differences to fail among the groups. So we decided we're probably just going to revert back to the original procedure with the next cohort to increase their numbers. However, it appears that CHAMP do have pretty typical balance and coordination and learn to increase their speed at the cadence that they're supposed to. Now this is where it gets pretty interesting. Acoustic startle and pre-pulse inhibition. So the acoustic startle is a well-studied measure of reactivity and sensor-motor integration. During a session, the mice are placed inside of this little tube thing, and they are stimulated with white noise bursts at several intensities. So I have the blocks here. They're loaded into the cylinder on a platform with electrodes to detect their vibration. So when they're in the tube, they kind of vibrate, and the electrode accounts for that, and that's what they measure. So these blocks are block A. You do 120 decibel white noise bursts. Now the reason why it's 120 white noise decibel bursts is because that is the mouse's startle response. That is the decibel where the mouse will get scared. So that is the highest decibel that it will go to. Block B is randomized trials of no stimulus, so no white noise, 100 decibels, 110 decibels, and 120 decibel, which is the startle response, white noise bursts to generate an input-output curve. Now the input-output curve shows us that the mice are able to hear properly and are able to go on to block C, which would be pre-pulse inhibition. It's randomized trials of pre-pulse stimuli intensities. So there'll be no pre-pulse, 78, 81, and 87 white noise bursts that precede the 120 decibel startle evoking burst. I will go over in much more detail about pre-pulse inhibition because it's a little complicated to explain in a later slide. And block D is again a 6 times 120 decibel white noise burst to compare with block A to see if they're able to habituate to that startle response. So for our input-output curve and our habituation, the males knockouts show decreased responses to all the intensities, while females show typical responses. However, they're all able to hear properly all of the responses that are shown. We can see that they're able to hear the white noise bursts and that they're both are able to go on to pre-pulse inhibition. For habituation, it's pretty variable, but there was not really any differences among the groups. Now, pre-pulse inhibition. The way I describe pre-pulse inhibition is, you know how when something like a noise is just there forever and at some point you kind of just ignore it? That's kind of what pre-pulse inhibition is. The pre-pulse is kind of like that noise, where you want to ignore the noise. You're supposed to ignore the noise, and it's supposed to prep you for a higher order noise. So for this pre-pulse, what it's supposed to do is you have the pre-pulse, and then a couple milliseconds later, they end up getting the 120 decibel burst, and we record their startle response. If their sensomotor gating is intact, they are able to not react to the 120 decibel startle burst as much as they would if they had a deficit. Now, sensomotor gating is basically the ability to perceive that the pre-pulse is kind of like an indicator to the louder pulse, to the startle pulse. So as you can see here, we did it in randomized trials of 78, 81, and 87. The higher the pre-pulse, the higher their percent inhibition to be able to ignore the 120 startle burst is. That's how it should be with wild types. However, the heterozygous CHAMP mice, you can see they have a deficit in that. They are not connecting between the pre-pulse is the indicator to the 120 startle burst. So they have it seems to be that they have some sort of sensomotor gating deficit, which is very interesting because that gives us things to look at, some physiological things to actually look for. So this is the brain stem where you're able to see where the acoustic startle response is well characterized. So basically, and these are all of the circuitries and neurotransmitters that are related to acoustic startle and pre-pulse inhibition. Basically what happens is you get the white noise, the loud startle. It goes into your eardrum. It goes through the chapitoid body, goes up to the giant PNC neuron, goes straight down the spine, and it goes into the motoneuron, and that's what characterizes the muscle movement of the vibration. Since we have this pre-pulse inhibition deficit, we are now currently looking at this this diagram and these neurotransmitters and these circuitries to see what kind of drugs and therapy we can use to target this specifically. So, for future directions, like I said, we'll be using gene therapy to restore CHAMP1 levels to reverse the acoustic startle. Like Dr. Pakai said, we bred mice. We injected the AAV9 vector into these mice. In 8 to 10 weeks, we are going to be doing acoustic startle to be able to see if we can reverse this deficit. Also, what I'm currently doing is using neurotransmitter-specific drugs to reverse this phenotype. So I am currently using haloperidol. It is a well-known medication that's been used for PTSD, ADHD, OCD to reverse their acoustic startle deficits. Some mouse models have used this as well to be able to reverse their pre-pulse inhibition deficits. So in a couple of weeks, I will be running that battery, injecting the mice, and seeing if it can be able to reverse their deficits. Lastly, I'm also currently doing this. We are doing learning and memory batteries. So novel object recognition, Y-maze, and fear conditioning. It is a 2-week process. Each cohort takes 2 weeks. Novel object recognition is long-term memory. Fear conditioning is working memory. So we will see if we are able to see if we can, with the regular CHAMP mice with no vector, with no drugs, see if there is an issue with the mice, and then hopefully be able to reverse that deficit later on. Thank you.
Any question? Thank you very much. Any question?
Many compliments, because a fine phenotypic analysis and characterization of knockout mice is essential to explore the effect of new therapeutics. However, my question is, in your opinion, how you think that this model can help the typical CHAMP1 disease that is associated with different gene variants, in particular with the truncating variant, and nowadays there is a consensus in not considering haploinsufficiency the underlying pathomechanisms. Anyway, your analysis is excellent, so I hope that can help in the future. Thank you.
I don't really, so I feel like we'll need more mice and more variants maybe, like of mice, maybe develop new mouse models that aren't just the heterozygous 35 base pair deletion. That could be really helpful, just so that we have more variants, because like you said, something might not help one mutation, but it can help another mutation, vice versa. So that would be very interesting. I don't know if, like you said, if the 35 base pair deletion would, show similar results compared to truncated or all of that. So we will have to see. Any other questions?
So thank you very much again.